Neurons, like all other cells, are going to have what we call a resting membrane potential. And what a resting membrane potential is it's a difference in charge between the inside of the cell membrane and the outside of the cell membrane. Note this membrane potential is called a membrane potential because it really does exist just across the cell membrane. Like all other cells, this is going to be negative. There's a wide range of charges that a neuron might have depending on type of neuron, but the average resting membrane potential of a neuron is about minus 70 millivolts. And what causes a membrane potential is going to be the difference in the concentration of ions between the inside and outside of the cell membrane, as well as a difference in permeability to those ions. If the cell membrane was completely permeable to the ions, then they just diffuse to their along their concentration gradients, and the inside would end up the same as the outside. Instead, the cell membrane, remember, it is going to be impermeable to ions except through ion channels. And so the cell can control how permeable it is to an ion and which ions it's permeable to. Remember, due to the action of sodium-potassium ATPase, also called the sodium-potassium pump, we are going to have a high concentration of sodium outside of the cell and a high concentration of potassium inside of the cell. Now, neurons are not normally very permeable to sodium, so that sodium stays outside of the cell. However, neurons are much more permeable to potassium due to the presence of a type of leak channel. So leak channels are these ion channels that are more or less open all of the time. So neurons are somewhat permeable to potassium. And since there's a high amount of potassium inside of the cell, that potassium is going to follow its chemical gradient and go outside of the cell. When that potassium leaves the cell, it's going to carry positive charges with it because potassium is going to be a cation. And that's what's going to actually generate most of that negative resting membrane potential. Neurons are excitable, which means that they can rapidly change their membrane potential in response to a stimulus. And this is going to be done by changing their permeability to ions to allow those ions to flow. And this is going to be done by opening up ion channels. So resting membrane potential, again, we have this sodium-potassium ATPase, or the sodium-potassium pump, which is going to pump 3 sodium out and 2 potassium in against the concentration gradient by expending ATP. And so we are going to have a high concentration of sodium outside of the cell and a high concentration of potassium inside of the cell. If we just had potassium channels, potassium would leak out and we would have a resting membrane potential of about minus 90. We are also a little bit permeable to sodium, so some sodium comes in, and that brings down the resting membrane potential to about minus 70. Because remember, sodium moving in makes you less negative because sodium has a positive charge. But because it's much more permeable to potassium than sodium, much more permeable to potassium than sodium, more potassium leaves the cell than sodium enters the cell, and so we stay at this resting membrane potential of about minus 70. Note our sodium-potassium ATPase pump is going to help maintain that gradient, but doesn't contribute a lot to the actual difference in charge. When we talk about electrical impulses and electrical signals, it's important to know some terms of electricity. So the basic terms that we're going to talk about is voltage, current, and resistance. Voltage is going to be a potential energy generated by a difference in charge, like we have across the membrane. So we are going to measure our membrane potential in voltage, specifically in a unit called millivolts. The more difference we have in charge, the more energy we have pushing charged particles from one end to another. So here we have voltage, and you can see the voltage is trying to push our particle through. The amount of charged particles that move is called current. So current is the actual flow of electrical charge between 2 points. In the case of our membrane potential, current literally means that ions move across the membrane through ion channels. So when we say current or flow, that's going to be the actual moving of ions like sodium and potassium across the cell membrane. And then we have resistance. And resistance is going to inhibit the flow of charged particles. And resistance is going to be measured in ohms. And if a substance resists the flow of charged particles, it's called an insulator. If it assists the flow of charged particles, it's called a conductor. So insulators have high resistance, conductors have low resistance. Ohm's law describes the relationship between these three factors as current is going to be voltage divided by resistance. So the larger your voltage and the smaller your resistance, the more current or flow of charged particles you're going to get. Ion channels are going to be responsible for allowing that flow of charged particles. Ions cannot pass directly through the cell membrane. Our cell membrane is a resistor. It is going to prevent the flow of those charged particles due to these hydrophobic fatty acid tails. Charged particles having a charge are hydrophilic. Our ion channels can be selective. They can allow only certain types of ions to actually flow through. And then we can divide ion channels up into leakage channels and gated channels. Leakage channels or non-gated channels are just always open. For example, our cell has a lot of potassium leakage channels, which makes it permeable to potassium. Gated channels are going to be channels that can open or close, and they can open or close in a number of different ways. For example, we can have chemically gated ion channels. A chemically gated ion channel attaches or opens when a specific chemical attaches. In our case, these chemicals are going to be neurotransmitters. Voltage-gated channels are going to open or close in response to changes in membrane potential. These are going to be responsible for forming and propagating action potentials. And then mechanically gated channels, these are going to be important in certain types of sensors. They actually open or close based off of stretch in the cell membrane. So, for example, our touch receptors have mechanically gated channels, which allows the cell to fire when the cell gets pressed on. Ions are going to diffuse according to their electrochemical gradient. So we've talked about this chemical part of the electrochemical gradient before. So this is just based off of concentration. Substances move from where they're at a high concentration to where they're at a low concentration. Potassium is high inside of the cell, so if you give it a path to move through an ion channel, it's going to move outside of the cell. However, they are also going to follow electrical gradients. So charged particles are going to be attracted to the opposite charge. Positively charged particles are attracted to negative charges. Negatively charged particles are attracted to positive charges. They're also going to be repelled by the same charge. Okay? At rest, the inside of the cell is negative. So it's going to attract cations and repel anions. So at our resting membrane potential, potassium is going to leak out through those leak channels, and as potassium leaks out, the inside of the cell membrane gets more and more negative. That's going to produce an attractive force that's actually going to pull potassium back towards the cell. So potassium keeps leaking out until enough potassium has leaked out that a strong enough negative charge is formed that's going to actually pull potassium back in. When the cell becomes stimulated, sodium channels are going to open, and sodium channels have both a chemical and electrical gradient that's going to force sodium in. Sodium both is at a high concentration outside of the cell, so it's going to want to move in, and also the inside of the cell is negative, sodium is positive, so it's going to be attracted. So when sodium-gated ion channels open, sodium is going to rush into the cell very, very quickly. That's how we're going to change the membrane potential. So the membrane potential is going to change when the membrane permeability to ions change. And that's going to happen when ion channels open or close. The opening or closing of the ion channels is going to change the concentration of ions across the membrane, and that's going to produce a change in the membrane potential. We call these changes in the membrane potential either graded potentials or action potentials. A graded potential is just a localized change in the potential across the membrane. This is typically going to be caused by gated ion channels, specifically chemically gated ion channels, specifically ones that are what we call neurotransmitter receptors. Ion channels that open in response to a neurotransmitter binding to them. Note these graded potentials can either be what we call excitatory or inhibitory. Excitatory potentials are going to make the cell excited, more likely to fire an action potential. Inhibitory potentials are going to make the cell less likely to fire an action potential. Action potentials are going to be these long distance signals that travel down axons, and they're actually going to be caused not by chemically gated ion channels, but by voltage gated ion channels. These changes in membrane potential are going to be used as the electrical signals that allow neurons to receive, integrate, and send information. So here, when we are looking at this change here, this is going to be caused by graded potentials. And then if we reach threshold, that's going to trigger the opening of voltage-gated ion channels, and that's what's going to cause an actual action potential to occur. This big change in the membrane potential that's going to sweep down the axon and actually send the signal. When we change the membrane potential, we can either do what's called depolarizing the membrane potential or hyperpolarizing the membrane potential. Remember, polarized means the two sides are different from each other. In our case of the resting membrane potential, the inside of the cell is more negative than the outside of the cell. So when we depolarize, we make the inside of the cell less negative. It moves towards zero. These are excitatory. So because again, if we depolarize to threshold, we're going to fire an action potential. If a membrane potential goes the other way, where we become more negative away from zero, that's called hyperpolarizing, making more different. And this is going to make the inside of the cell membrane more negative, farther away from zero, and it's going to be inhibitory. decrease the likelihood of generating an action potential.